let's begin with this thing of M phase. So it is the M phase is the most dramatic period of the cell cycle. We otherwise call this as dividing phase. Coming to the M phase, it involves a major reorganization of virtually all components of cell. Since the number of chromosomes in the parent and progeny cells is the same, it is also called as equational division, because if assume that a cell is starting with 46 chromosomes, the daughter cells also will be having 46 chromosomes. Though for convenience, mitosis has been divided into 4 stages of nuclear division, that is karyokinesis. It is very essential to understand that cell division is a progressive process and very clear cut lines cannot be drawn between the various stages. So karyokinesis involves 4 stages: prophase, metaphase, anaphase, telophase. Remember the mnemonic PMAT. So coming to the very first thing, prophase, which is the very first stage of karyokinesis of mitosis, follows the S and G1. After following the checkpoint of S and G2, it enters into prophase. So in the S and G2, the new DNA molecules formed are not distinct but intertwined. So prophase is marked by the initiation of condensation of chromosomal material. So the chromatin material is condensed to form chromosomes. The chromosomal becomes untangled during the process of chromatin condensation. The centrosome, which had undergone the duplication during S phase of interphase, now begin to move towards the opposite poles of the cell. The completion of prophase can thus marked by the following characteristic events. One is the chromosomal material condenses to form compact mitotic chromosomes, and chromosomes are seen to be composed of 2 chromatids attached together at the centromere. So initially the cell will be having the nucleus. Inside the nucleus you will be having the chromatin material like this. So we'll see with the other color. We'll be having like this. Now what it is happened in the prophase? Now inside the nucleus, the chromosomes are being formed with 2 chromatids. Okay, attached at the centromere. Centrosome, which has undergone duplication during interphase, begins to move towards the opposite poles of the cell. So each centrosome radiates out the microtubules, which we call it as asters. So 2 asters together with spindle fibers forms the mitotic apparatus. So you will be having the cell, nucleus with the chromosomes, and 2 spindle fibers, 2 centrosomes go to the opposite poles from here. So the microtubules will be coming like a star. We call that as asters. Cell at the end of prophase, which means the late prophase. When viewed under the microscope, do not show Golgi complexes, endoplasmic reticulum, nucleolus and nuclear envelope. So this is early prophase. In the early prophase, you can see the untangling of this chromatin material to form as chromosomes. So like this, the chromosomes are being formed with the centromere, and the degeneration of nuclear membrane is started. It is late prophase. So entirely the endoplasmic reticulum, Golgi bodies, entirely it got invisible, and nuclear membrane is also degenerated. So coming to the next thing, metaphase. The complete disintegration of the nuclear envelope marks the start of second phase of the mitosis. Hence, the chromosomes are separated through the cytoplasm of the cell. By this stage, the condensation of chromosomes is completed. They can now observe clearly under the microscope. And this is the stage which morphology of chromosomes is most easily studied. At this stage, metaphase chromosomes are made up to sister chromatids. You can see here the relationship between this and this chromatid is sister chromatids. But the relationship between this and this is non-sister chromatids. When it is held together at the centrosome, small disc-like structure from the surface of the centrosome, we call it as kinetochore. So this is the kinetochore. To this, the aster, the spindle fibers are attached. So this structure serves as a attachment of the spindle fiber. So to the chromosome that is moved into position at the center of the cell. Hence the metaphase is characterized by all chromosomes having to lie at the equator of one chromatid of each chromosome connected at its kinetochore to spindle fibers from one pole, and its sister chromatids connected by its kinetochore to spindle fiber from the another pole. So one pole is connecting through the kinetochore this sister chromatid. Other pole is connecting the kinetochore of this sister chromatid. Now this plane of alignment of chromosomes at the metaphase is referred to as metaphase plate. The key features of metaphase are spindle fibers are attached to the kinetochore of the chromosomes, and chromosomes are moved to the spindle equator and get aligned along the metaphyseal plate through the spindle fibers to both the poles. And anaphase. So this is metaphase, transition to metaphase, from late prophase to transition to metaphase, and this is the entire metaphase. The center, this we call it as metaphyseal plate or equatorial plate, and the center alignment. During metaphase only the best study the size and shape and alignment of the chromosomes. Onset of anaphase, each chromosome are arranged at metaphyseal plate, is split simultaneously, and two daughter chromatids, now referred to as daughter chromosomes, of the future daughter nuclei. So begin their migration towards their two opposite poles. As each chromosome moves away from the equatorial plate, the centromere of each chromosome remains directed towards the pole, and hence at the leading edge, with the arms of the chromosomes trailing behind. So thus the anaphase is characterized by following key events. So the centromere split and chromatids got separated. Chromatids moved to opposite poles. So these are the two things that is been happening. And finally we'll come to telophase. Telophase is actually the reverse of prophase. In the beginning of the final stage of karyokinesis, that is telophase, the chromosomes that have reached their receptive poles decondense. Means the condensation of the chromosomes, now it is decondensing to form the chromatin material and lose their individuality. The individual chromosomes can no longer be seen, and each set of chromatin material tends to collect at the end of the two poles. This is the stage which shows the following key events. Chromosomal cluster at the opposite spindle poles, and their identity is lost as discrete elements. And nuclear envelope nucleolus is reformed. So develop around the chromosome at each pole, forming two daughter nuclei. So the nucleolus Golgi complex and ER is reformed. Whatever is been absent, whatever is been happened in the prophase, the entire reverse thing that is going to be happen in telophase. So this is about the PMAT. So remember one mnemonic. Pro means first. So what are the first things that is going to be happened? and M for middle. So the middle alignment of chromosomes in the metaphyseal plate. A for away. The splitting of centromere to separate the sister chromatids and to form separate daughter nuclei, separate daughter chromosomes. And telophase 2. Two separate daughter nuclei is formed. So this is the mnemonic for mitotic phase, which is dividing phase. So thank you.